at that point, I really was curious on how are other animals distinguishing the sex of their conspecific. So I went into the literature and investigated a little bit what people had described in other species. And here's what I found. So in this particular species of bird, the shell parakeet, this is a female, and this is a male. And the animal, the parakeets, recognize the gender of their conspecific based on the presence of these blue dots on the top of the beak. And this particular blue dot is essential for sex identification, which is that if you paint a blue dot on the beak of a female, the animal now is identified as a male, and other males will attack that female with a blue dot, thinking it is a male. And similarly, if you mask the blue dot from the beak of a male, then the other males are going to try to mate with this male without a blue dot by thinking it's a female. So the blue dot, the visual cues, the identification of this blue dot is essential to the identification of this animal as a male or female. Similarly, in this other species of bird, the American flicker, he is a female, he is a male, and what provides the identification of one being male, the other one a female, is the presence of the black mustache. So if you were to mask the black mustache on the male, this animal will be identified as a female, and the other males will try to attempt to copulate with this male without a mustache. And this animal here that is a female, if you paint a black mustache, males will attack that female thinking it's a male. And Timbergen, who is a very famous ethologist, described this by calling those signs the badges of masculinity, which is here, this is my blue dot, or this is my mustache, I'm a male, and if I don't, then I'm a female. And so this is quite interesting, is a visual recognition of the gender identity. And I think that what we found for the pheromones is the olfactory equivalent of the badge of masculinity, which is the voronasal organ is responsible for discriminating between males and females. Now, obviously, the animal we care the most about are humans. What kind of strategy do humans use? And is the voronasal organ also being used for sex identification? And here things are likely to function very differently. The TRPC2 channel, which is responsible for the function of the voronasal organ in rodents, is a non-functional gene in humans and actually in higher primates. These little triangle that you see here, there are 9 of them, are the site of deleterious mutation, such that higher primates and humans have a number of deletion or frame shift or nonsense mutation that make this gene unable to generate a functional protein. And it's quite interesting to actually look throughout evolution when this mutation occurred, and this is the work of the laboratory of Emily Lyman, that shows that the mutation really start to accumulate at the split between New World monkeys and Old World monkeys and apes. So all this part of the tree of higher primates really is unlikely to use the voronasal organ as a tool to discriminate sex. And I think what's quite interesting and was proposed by Emily Lyman's group is that these split here between the New World and the Old World monkeys also correspond to the duplication of the red and the green opsin genes, such that animals in this part of the tree here have an additional opsin receptor gene, and therefore the ability now to discriminate between 2 colors, red and green, whereas animals in this part of the tree have one opsin gene that detect both green and red color